Hello students, my name is Praveen Kumar, lecturer in zoology. Now we are going to discuss about zoology model paper 2. So in this model papers, the first question is thecodont dentition and diphyodont dentition. So what is thecodont dentition? You know, if you see the jaw socket, okay, so our mammalian jaws are with sockets. So in these sockets, teeth are embedded. You see this red color what I am mentioning here, you know these are the teeth. So where they are? They are in the sockets. So what is the definition you will give for these? Teeth which are present or which are embedded in the sockets of jaw is called thecodont dentition. So especially in which animals you can see this type of thecodont dentition you can observe in the mammals. The best example is human being. And the next one we are going to see, another one is diphyodont dentition. What is diphyodont dentition? In the diphyodont, the name itself says that di means two. So means in our lifetime we have two sets of teeth. One is milk set and another one is permanent set. So like that, milk set and permanent set, there is two types of teeth are present in our body. Especially in the childhood we have milk teeth. They are replaced by permanent teeth. So that's why in the lifetime there is two sets of teeth. So that's why it is called diphyodont dentition. Okay, understood now? Okay students, let us discuss question number 2. So here you see the question, this is rennin and rennin. What is the difference? Here you can see the double N, here you can see the single N. The pronunciation is same, rennin, rennin. So this type of question is very important. So you see where you can see this rennin. Generally this rennin is present in the stomach, especially in the young children, okay, who is suckling the mother milk. To convert the milk, so first point here it is present in the stomach, okay, especially in the stomach of infants. Okay, what is its use? It is useful to convert the milk to curd. Milk is converted into curd. Means here in milk there is a protein called casein. What is that? Casein is a milk protein. This casein is converted with the help of calcium ions. It is converted into calcium paracasein. So like that, casein is converted into calcium paracasein with the help of rennin in the stomach of infant babies. That's why generally we will observe, if you see the small children, after drinking milk, after some time immediately they will vomit curd. That is the secret behind the rennin. Okay, let us see the next one. This is renin, rennin. So where you can see it is released from the kidney, especially juxtaglomerular cells. What are that? Juxtaglomerular cells. Where the juxtaglomerular cells are there? They are present in the kidney. So especially in the nephron structure we can see these JG cells. What is the use of this? They can able to release enzyme rennin. It is also called angiotensinogen, which is also called angiotensinogen, which is going to liver. From where it is released? It is released from JG cells, and where it is going? It is going to liver. What it is going to do in the liver? In the liver, it is going to convert angiotensinogen is converted into angiotensin 1. So here it is also inactive, and whatever the product is formed due to the action of this rennin, this is also inactive. So like that, even inactive is converted into another inactive form. That is happens because of rennin. Okay, let us see the question number 3. So in this question number 3, they asked about motor unit. Okay, so what is motor unit in relation with muscle and nerve? Muscles and nerve. So in this you can see, suppose you see this is all about muscle. This is all about muscle. So into this muscle, a telodendrites of a neuron. What is this neuron? Motor neuron. What is this? Motor neuron. So what are these? Telodendrites. You see in this diagram, motor neuron and what are these? Telodendrites. So motor neurons, telodendrites are innervated with the bundle. Which bundle it is? Muscular bundle. So that's why whatever the messages are coming through the motor neuron, they are going to given for the particular muscle. So like that, the telodendrites which are innervated into the bundle of muscle fibers is together called motor unit. It is very important for the activation of muscle bundle for the contraction purpose. So like that, it is a motor unit in concern with muscles and nerve. Okay students, let us discuss the question number 4. So what is this question? What is anti-diuretic hormone? What is this? Anti-diuretic hormone. So in this anti-diuretic hormone, it is also called ADH. That is anti-diuretic hormone. Officially its name is vasopressin. What is this? Vasopressin. From where it is released? It is released through posterior pituitary gland. You know pituitary gland. This part is posterior pituitary gland. Posterior pituitary gland. So from the posterior pituitary gland, the vasopressin is released. Okay, actually it is synthesized in the hypothalamus and it is released through posterior pituitary. Okay, what this is going to do? Vasopressin, it is going to act on the distal convoluted tubule of nephron. Where it is acting? It is going and working on the DCT, distal convoluted tubule. For what purpose? To absorb water. So especially reabsorption of water. Reabsorption of water from the distal convoluted tube. So automatically water is reabsorbed, automatically the loss of urine. Okay, so here loss of water through urine is prevented. Loss of water through urine is prevented. So that's why concentrated urine released out. So it prevents diuresis. What is the meaning of diuresis? Loss of water through urine is called diuresis. So this hormone is, by absorbing water, it is preventing the loss of water from our body. So that's why it will help us to prevent dehydration. So due to the lack of this hormone, the disease will come, that is called diabetes insipidus. Diabetes insipidus. So like that. Okay, from where it is releasing and which work it is going to do, and if the Okay dear students, let us discuss the question number 5. So in this question they asked about what are autoimmune disorders. Autoimmune disorder. Okay, so what is the meaning of autoimmune disorder and what are the examples? So first you see auto. Auto means self. Immune means automatically which is giving disease resistance power to our body. So here in this mechanism you can see our antibodies of our immune system, they are failed. What happened? Okay, antibodies of our immune system are failed to recognize some of our proteins. They feels that these are foreign proteins. Actually, our self proteins are treated like foreign proteins. So that's why these failed antibodies in recognizing the particular self and non-selves, so they starts attacking self proteins. So like that, that's why immune system is underwent disorder. So like that, autoimmune disorder comes by the failure of antibodies in recognition of self proteins. They recognizes them as a foreign, so that's why they attacks. So this type of disorder is called autoimmune disorder. So what are the examples? Here the examples are Graves' disease. Generally it is going to attack thyroid gland. So that's why Graves' disease, and another one is rheumatoid arthritis. Rheumatoid arthritis. So like that, Graves' disease and rheumatoid arthritis. Rheumatoid arthritis means generally, okay, lot of synovial fluid accumulated in the joints. So like say joints get swelling. So because of the attack of these particular antibodies, lot of fluid is secreted. Here Graves' disease, okay, lot of thyroxine is secreted. So anyway, Graves' disease and rheumatoid arthritis are the some examples for this particular autoimmune disorders. Okay students, let us discuss the question number 6. In this question, okay, the diagrammatical question it is. So draw the diagram of sperm. So you know sperm consists 3 parts. Okay, so this is part 1. Okay, and this is part 2. Okay, so and this is a tail. So like that, you see the students here in this diagram, the region which is called head region, this region is called middle piece. Okay, middle piece, and the third region is tail. So like that you have to draw the border. So what you have to mention, in the head region there is a nucleus. So haploid nucleus is present. So that's why you must mention the nucleus. And the tip. So in this tip, you know, there is a acrosine, acrosome or acrosine enzyme is present. So and the middle piece, what is the specialty of the middle piece? In the middle piece there is many number of mitochondria are present. What are these? Many number of mitochondria are present, which helps okay, in the swimming of sperm. Okay, you know this next part is okay, tail region, which helps for swimming. So like that, these parts you have to mention clearly, so you will get full marks. And in between this middle piece and head, you can put a small depth, okay, give it as a neck like that. So this is all about sperm diagram. And the outer coat, if you are interested to draw the outer coat also, you can mention the outer coat as a plasma membrane. What is this? Plasma membrane. Okay, you can draw the diagram neatly. You will get 2 marks. Let us see the question number 7. So here in this, it is given amniocentesis. Okay, amniocentesis. So what is the amniocentesis and what is its use? So amniocentesis means, you know, if you see a pregnant woman, so in this pregnant woman, you know, this is the okay, placenta and developing fetus you are observing in her uterus. Okay, so the developing baby, around the developing baby, you know that there is a fluid. What is this fluid? This fluid is called amniotic fluid. Amniotic fluid. So this amniotic fluid will consist some of the cells of epiblast. So that's why what the doctor is doing to detect, or we can also call to diagnose the genetical disorders. So okay, doctor collects this amniotic fluid. So around the baby amniotic fluid is there. That fluid is collected by the specialist doctor, okay, to detect or to diagnose the genetical disorders of unborn baby, genetical disorders of unborn baby. So what type of disorders? Especially Down's syndrome. What is this? Down's syndrome and Turner's syndrome. Turner's syndrome. Okay, and Edwards syndrome. Edwards syndrome. So like that. Okay, what is there any problem to the fetus? Especially genetic disorders are detected by collecting amniotic fluid. In that amniotic fluid, epiblast cells are there. From that cells, they can detect the chromosomal disorders. What are the disorders? Down syndrome, Turner syndrome, Edwards syndrome. Okay students, let us discuss question number 8, which is from genetics, that is Klinefelter syndrome. What is this? Klinefelter syndrome. What is Klinefelter syndrome? Generally in the Klinefelter syndrome, okay, 47 chromosomes are there. Actually how many should be present in a human being? 46. But here how many? 47. Means one chromosome is extra. Which chromosome is extra? Let us see. Generally, autosomes are 44. What is this? Autosomes are 44. What about the allosomes? They must be XY. But here, one chromosome is extra. So, that's why you can see XXY condition. So, the 23rd pair, that is allosome. 23rd pair shows trisomy. Because of this, okay, they shows 47 chromosomes. One extra X chromosome is there. Instead of XY, XXY you are observing. So because of this extra X chromosome, female characters are not suppressed in males. Female characters are not suppressed in males. So that's why they behave like females, even though they are males with Y chromosome, but due to extra X chromosome, they behave like female. Their characters are like female, and they shows bar body. Okay, in their chromosomal composition, they shows bar body, and also gynecomastia. Gynecomastia means slight development of breast also we can see, and there is no growth in the reproductive organs of, especially in this male. This type of special condition is called Klinefelter syndrome. Let us discuss 9th VSAQ. So in this 9th VSAQ, this is from evolution. You can see what is genetic load. What is genetic load? So give one example. Generally, continuation or existence of deleterious genes. Existence of deleterious genes in the population. Existence of deleterious genes in the populations is called genetic load. Delete word, we are using delete word, means what? Compulsory that animals, that type of animals which are dangerous genes with these type of animals should not continue their life in our population. But some genes which are very dangerous, they are continuing, they are existing in the form of heterozygous condition. In the form of heterozygous condition, they are surviving in the nature. For example, if you see sickle cell anemia, sickle cell anemia, in the sickle cell anemia, if you see this HbA, this is a normal people's genome. Now you see HbA, HbS. So this is heterozygous condition, and another one is HbS. So in this HbS and HbS, these people are going to die. So why? Because they will not have proper red blood cells, especially the formation, structural formation is defected due to this particular deleterious gene. So the people will die in homozygous condition. But in heterozygous condition, these people, whatever the dangerous gene is there, that is HbS, is surviving. So that's why these people, this type of dangerous gene is continuing in the population, and they have particular character, they are resistant to malaria disease also. So like that, genetic load means existence of deleterious genes in the population. The example is sickle cell anemia. Okay students, let us see question number 10. This is guardian angel of cell genome. Okay, guardian angel of cell genome. So what is this? We will see now. Generally, okay, if you see the cell cycle, there is a G1 phase, next S phase, and next G2 phase, and next M phase. Generally, you know very well about these cell division stages, especially in the G1 stage. Okay, so the guardian angel of genome will work. This is called P53 protein. What is the name of this protein? P53 protein. This P53 protein is going to check the ending stage or the climax stage of G1. What is this? G1 stage is going to get end. At that time, P53 protein will work. Especially, is there any repair needed to DNA? Okay, so what it is going to do? It is going to repair the DNA. So that's why the perfect DNA will pass to next stage. If the repair is not there, if DNA is not corrected, automatically what happens in S2 stage? Same DNA with mistakes is going to get another copy in the S stage. So that's why S stage is very important for the replication of DNA. That's why DNA is once checked before entering into S phase by the P3, that is P53 protein. So that's why it is called guardian angel of our body. It prevents cancers. It prevents cancer, especially by controlling the cell division mechanism.